So, before we go into case studies, let us discuss some topics on something that is very important and emerging in modern drug discovery, which is the rampant observation of drug resistance. So, here in today's lecture, we will look at both concepts of drug resistance as well as drug synergy. Okay. So, drug resistance is observed when a formerly effective drug dose is no longer effective. So, this could be a natural resistance or an acquired resistance. Resistance mainly arises by natural selection. So, for example, a drug may destroy all the organisms in a colony. So, if you have a bacteria like in a 10 million organisms, one organism will survive and the rest of them will die. But that one organism can survive and divide and perhaps have grow into a new colony, right. So, that one organism which was naturally resistant to the antibiotic or the drug is what is going to be causing this drug resistance. Okay. So, it is likely that this organism has one or more mutations that make it resistant to the drug. So, once all the susceptible organisms have been killed, a few resistant one can replicate and eventually become the predominant species. So, bacteria may also resist antibiotics as a result of chromosomal mutations or inductive expression of a latent chromosomal gene or by horizontal gene transfer, that is exchange of genetic material through transformation, transduction or conjugation by plasmids. So, these plasmids are nothing but small pieces of circular DNA that can carry genes for survival of the organism. And if there is a group of organisms which are already resistant to the antibiotic, those genes can be transferred to another organism and that helps in the other organism to become resistant to the antibiotic. So, because these drugs are rarely mutagenic, therefore, resistance by drug induced mutation is a very rare situation. So, because of the remarkable ability for microorganisms to evolve and adapt, there is a need for new drugs with new mechanisms of action that are not susceptible to the existing or prevailing mechanisms of resistance. So, this is a major area of drug discovery that is going on right now, including in my lab. So, much like bacteria, you also have cancers or tumors which have similar considerations. So, once we expose the tumor to a drug, then it is possible that a large number of those actually are killed, but some of them which have existing mutations which can help with resistance are likely to predominate. Now, then these will evolve and adapt and to ensure the survival of the cell lineage. Antibiotic resistance is now such a huge problem across the world that it is quite likely that in the next decade or two that even simple infections will be difficult to cure. Similarly, resistance to anti-tumor drugs is becoming a huge problem. So, the mechanisms of drug resistance are classified into two major classes. The first one is exogenous resistance, is when new proteins are developed by the organism or cell to protect it from the drug. The other way to develop resistance is by endogenous resistance. This occurs by mutation or even single point mutation, that is one amino acid changes in a protein and that can result in resistance. So, now let us look at each of these in fair bit of detail. So, the first one is altered target enzyme or receptor. Here mutation of the amino acid occurs. So, here the important amino acid that was originally present in the active site has now been replaced by another amino acid. So, once you have the binding site, so here is your drug and here is the binding interaction. Now, it is possible that one of these binding interactions becomes weaker, right. So, let us say the mutation occurs here and this becomes weaker. Now, if this becomes weaker, then the dissociation can actually predominate and therefore, the efficacy of the drug goes down. You can also have allosteric modulation where mutations elsewhere in the conformation. So, let us say you have a protein. So, the mutation can occur here and this results in conformational change which changes the binding pocket of the enzyme. So, this is now this is not very good at binding anymore. For example, resistance to the antibiotic trimethoprim derives from a single mutation of a single amino acid of the target enzyme, which is dihydrofolate reductase. So, the properties of the singly mutated enzyme differ somewhat from those of the normal enzyme, but it still binds to the substrate dihydrofolate. So, Now, the next way in which you can have resistance is overproduction of the target enzyme or receptor. So, let us say we have a excellent reversible inhibitor of an enzyme or an agonist for a receptor. Now, if you have a large number of these enzymes, that is if the protein expression goes up, then it is you need a much higher concentration of the inhibitor to actually go and inhibit the enzyme. So, one way in which the cell can counter this drug is to increase the enzyme production by induction of extra copies of the gene encoding the enzyme. Okay. So, for example, the enzyme dihydrofolate reductase by malarial parasite, the inhibitors of this has overproduction of that enzyme and in the unaltered form, and this results in resistance. You can also have overproduction of the substrate or ligand for the target protein. So, like we discussed earlier, a number of the inhibitors that we develop are reversible inhibitors. So, therefore, if the substrate concentration goes up, the efficacy of the inhibitor goes down. Therefore, this could be one way in which the organism is able to acquire or to become resistant. So, an example here is overproduction of para-aminobenzoic acid, which is the substrate for the enzyme, and sulfur drugs act on this enzyme, and therefore, by increasing the concentration of para-aminobenzoic acid, the bacterium becomes resistant to sulfur drug. You can also have increased drug destroying mechanisms. So, here in when you have rapidly dividing tumors or microbial cells, then they want to survive, and so they would upregulate these drug destroying mechanisms as a way to fend off the drugs that are trying to destroy them. So, this is something that again can occur in these organisms. So, resistance can occur by induction of genes to produce new enzymes, increase the quantities of existing enzymes, or produce other substances to degrade or sequester the drug. So, all of these may result in destroying the drug. So, within 10 years of introduction of penicillin, for example, half of the strains of Staphylococcus aureus had become resistant, and by 1990s more than 90% of the strains were resistant. The principal cause of resistance to penicillin is the generation of this enzyme called as beta lactamase. Here beta lactamase destroy the penicillin and they make the drug ineffective. We have looked at in detail about prodrugs, and prodrugs, the concept in prodrug is that you need metabolism for the prodrug to be activated. So, one other form of resistance is that if the enzyme that is activating the prodrug is going down in quantity. So, the activity of the enzyme can go down, and then that can result in drug resistance. So, an example here is the resistance to 6-mercaptopurine, which occurs by deletion of hypoxanthine guanine phosphoribosyl transferase, the enzyme which converts this ribosyl form to the hydrogen R equals H. Another major way in which resistance can occur is activation of new pathways that circumvent the drug effect. So, what this means is that if you can have the drug affecting a particular production or a particular enzyme, then the organism could bypass this effect of the drug by inducing a new metabolic pathway. Okay. So, let us say you have A going to B, going to C, and so on, and now if you inhibit this enzyme here, then it is possible that the organism bypasses this enzyme and finds a different way to carry out the same metabolic transformation. So, here is an example. So, N-phosphonyl L-aspartate was very successful in treating cancer, but it failed in clinical trials. So, this PALA, as this is called, it inhibits the enzyme aspartate transcarbamylase, okay, and this is very important in the de novo pyrimidine synthesis. So, when tumor cells acquired the ability to utilize preformed circulating pyrimidine nucleosides, then they no longer needed to have a de novo metabolic pathway for pyrimidines. So, therefore, this drug becomes ineffective. So, another way in which resistance can occur is to have altered drug distribution to the site of action. So, this happens when the cell or cellular organism excludes the drug from the site of action by preventing cellular uptake of the drug. So, one of the ways in which this can occur is by changing the net charge on the membrane. So, if you can for example, if there is a phosphatidyl glycerol membrane, it can be changed to lysophosphatidyl glycerol groups, then it converts an anionic group to a cationic group. So, this change can affect the permeability of the drug. So, if the permeability of the drug goes down, then clearly its efficacy will go down. In this way, the drug with the same charge can actually now be repelled. So, aminoglycoside antibiotics, for example, become ineffective because the quinones that mediate the drug transport, or from lack of quinones that mediate the drug transport, or from the lack of an electrical potential gradient required to drive the drug across the bacterial membrane. So, these two can result in decreased uptake of aminoglycosides, which is going to result in resistance. There are also what are known as efflux pumps, which are commonly expressed in many of these highly resistant cells. Efflux pumps are located basically on the protein surface, and what they do is that they pull stuff out from the cell and kick it out. Okay. So, if a drug gets in, but if the efflux pumps are very active, then the drug is going to get in here, but it will be kicked out. So, efflux pumps are a major problem in drug resistance. So, some transporters, which are known as multi-drug resistance pumps, are quite broad in their specificity, and they can efflux a variety of natural and synthetic drugs. So, both specific and broad specificity efflux transporter systems have been identified that contribute to bacterial resistance, and not just one or two, but these have a really broad range of antibiotics to which it is resistant to. So, these now become targets for us to develop new antibiotics. The next topic that we are going to look at is drug synergy. So, we have already looked at a part of this when we looked at drug alliances, but here we are just going to look at it a little bit more formally. So, drug synergy, when two drugs are given in combination, they can be antagonists, that means that one drug is going to cancel out the effect of the other, or it can be sub-additive, that means that there is going to be a little bit of addition in efficacy, or it can be additive, that means that the effect of A can be added to the effect of B, and that is going to result in the combination therapy. But more importantly, what we look for is synergy. So, here the synergy refers to when the therapeutic effect of these two drugs or more used in combination is greater than the sum of the effects of the drugs administered individually. So, the difference between additive and synergy is that when I add these two drugs at the concentration at which they are efficacious, the net effect does not go up, but in synergy the sum of the effects is larger than the individual drugs. So, one can think about a synergy by inhibiting a drug destroying enzyme. So, we have already looked at this previously. So, for example, you can have an inhibitor of enzyme that destroys the drug, and this will result in synergy. This particular molecule may not have any therapeutic effect on its own, but it only protects the drug from being destroyed. So, this is also called sensitizing the drug to, or sensitizing the pathogen to the drug. But if the primary drug and the inhibitor of the drug destroying enzymes are administered together, then together they can have a synergistic effect. So, we have already seen this previously in the case of ampicillin and clavulinate. So, they are administered together so that you can protect ampicillin from beta lactamases. You can also do what is known as sequential blocking. So, here when we look at a metabolic pathway that is important and we want to develop inhibitors. So, here is what we look at, let us say A goes to B, goes to C, goes to D, goes to E. So, this is a metabolic pathway that we want to inhibit. So, let us say we develop an inhibitor for this step. Okay. So, here because B does not get converted to C by inhibition, you do not get to the formation of E, right. But if less than 100%, if of course, because we are looking at reversible inhibition, it is possible that you would never get 100% inhibition. So, if then less than 100% inhibition of the enzyme activity is blocked, then the metabolic pathway has really not truly been shut down. There is always going to be some amount of E that is produced. But when you use inhibitors of two consecutive enzymes in the pathway. So, here what we would do is in this A going to B, going to C, going to D, going to E, what we would do is we would not just block this, but perhaps you would also block this. Okay. So, here you would develop two different inhibitors, so that they can work in synergy. We have already looked at combinations of drugs that we have previously discussed this in detail. So, I am not going to take this up again. As I mentioned earlier, efflux pumps are a major problem in not just in cancers, but also in bacteria, and these can act a very broad spectrum pumps which can kick out many drugs. So, what a large number of groups are now working on is to develop inhibitors for these efflux pumps. So, once you have an inhibitor for this efflux pump, then the concentration of the drug inside the cell. So, let us say you have a cell and you have your efflux pump. So, the drug gets in, but it is effluxed out. Okay. So, here if you inhibit this process, then the concentration of the drug goes up, so that it can hit the target of choice. So, using two different types of inhibitors for the same target can also be used. So, here is the example. So, you have mycophenolic acid and mesoribine, which both are inhibitors of inosine 5 monophosphate dehydrogenase, which is IMPDH. Right. So, mycophenolic acid has been shown to be an uncompetitive inhibitor of this enzyme. And on the basis of X-ray crystallographic studies, the complex of mesoribine monophosphate with IMPDH is proposed to mimic the tetrahedral intermediate or the transition state leading to it. So, this is an uncompetitive inhibitor, while this is a transition state inhibitor. Okay. So, therefore, these two inhibitors have different mechanisms of action, and so if we can give a combination of these two drugs, then we would expect it to be synergistic, and indeed it is.